Welcome everyone. We'll go ahead and get started. For those of you who don't know me, I'm Kate. I'm the scientific director of the foundation, and welcome back to the scientific sessions of the We Are Family Conference. Today we have a clinical trial session for you, and we have Dr. Berry Kravis joining us. I will actually give a short talk of about 15 minutes, and then she will give a talk, and we will save questions until the end. Just a reminder to put any questions into the Q&A and not the chat, because it's easier for us to monitor there, and to not ask any really specific particulars about your child or about specific medical advice, but more general questions. And we will go ahead and get started. So I will first share a brief word from our sponsor. So 1 second while I pull that up.
Our mission at Jaguar is to accelerate breakthroughs in gene therapy for patients with severe genetic diseases.
With gene therapy, we can give the opportunity at a better life. That's what makes Jaguar special.
All right, so I will just get started with this first presentation here. This should be really short, just 15 minutes, and I'm hoping that I can share some information that is relevant for both folks that aren't as familiar with clinical trials in Phelan-McDermid syndrome and those who are very familiar by sharing some more recent resources and information that we have. So how does a clinical trial in PMS get launched in the first place? There's a number of ways this can happen. First, oftentimes a pharmaceutical company is interested in a disorder such as PMS, either rather specifically or interested in a more general group of disorders such as autism or other neurodevelopmental disorders, and will be developing a drug for that group. And part of our role is to provide them with any information that's necessary for them to understand if our population is a good population for their drug. And that includes data from the International Registry and Data Hub and natural history studies such as genetics of our group, roughly how many people we have, what types of medications people are on. And that's really helpful for the pharmaceutical company deciding if they want to test the drug. And we are really thankful for everyone who participates in these things because we're able to give that information. So that's for drugs that are being developed. And there's also the possibility that existing drugs, which have already been either FDA approved or approved by the agency in a certain country for another disorder, can then be tested in Phelan-McDermid syndrome. And this is called repurposing drugs. And the majority of clinical trials that have already taken place in Phelan-McDermid syndrome have been repurposed drugs, but more recently we are trending towards newly developed drugs as well. So clinical trials are really one of the last stages of clinical research, and there's a lot that happens before them. First, there has to be some drug discovery, and this typically involves studying, pathways or proteins involved in a certain disorder and figuring out a way to target them. And this can be done in a lab in academia or in a pharmaceutical company. And then after that, it can be tested in animal models, ranging anywhere from fruit flies all the way up to non-human primates. And this is usually testing for safety and effectiveness. And if that goes well, it can then go into clinical trials where the group who is developing the drug or testing the drug would apply to start a clinical trial with a regulatory agency like the FDA. And the first phase is usually very focused on safety and dosage and has a small number of people. After phase 1, it can go to phase 2, which includes more people, and typically it looks at efficacy or effectiveness at fixing certain symptoms and side effects or adverse events. And this is not going to be several hundred people in Phelan-McDermid syndrome. This is probably closer to about 20 people in Phelan-McDermid syndrome in a phase 2, just because it is a rare disorder. After that, it can go to phase 3, where it's more of the same, but on larger number of people, and you're testing more long-term for safety, effectiveness. And one thing that I want to mention is that going from animal models to phase 1, there are challenges with modeling disorders sometimes, and there are cases when drugs are very effective in animal models and not as much in humans due to a number of reasons, including our complex biology and varied genetics and things like that. And this is not to be discouraging, it's just to be a reminder that clinical trials are always an experiment. And so it's not a finalized drug that is definitely going through until the end. And the last thing I want to mention is just that the time frame, it can take a while for this process. So once a drug goes into clinical trial phases, progressing through those phases can take years, and it can also take years leading up to the clinical trial. Once something goes past phase 3, they can apply to go to market. So this would include review of things like the labeling and the marketing and the production of the drug. And once it goes to market, even once people start using it, there is a prolonged continued phase of monitoring a large group of people for safety and effectiveness. And this whole process, again, can take months to years. There's a lot of terminology associated with clinical trials, and I don't want to go too much into this because I did make a clinical trials informational YouTube video, including some of this information, and I have some new information today. But there are terms in there such as inclusion and exclusion criteria, control groups, double-blind studies, all of these terms that feel free to go and check out that video and learn more about it, but didn't want to spend too much of your time on it today. There are a couple of other terms that I really want to highlight today that I didn't in that video. One of them is outcome measures. An outcome measure is essentially a test designed to see if the treatment is working. So in Phelan-McDermid syndrome, this could be something like behavioral tests or an EEG, or things that can actually measure, is there an improvement from the drug? And this is kind of an unusual choice, but I want to highlight a phrase called statistically significant. This is true of any study, whether it's a clinical trial or more basic research, but scientific publications are usually published with results on whether or not they are statistically significant. And this simply means that statistics have been applied that can say, beyond a certain doubt, that a effect of a drug or a treatment or something like that is unlikely to be due to chance. And there's some calculations that go into that. But I just wanted to highlight that if you're ever reading a scientific paper and you find, that there are some results that are statistically significant, especially from a clinical trial, just look at what they're testing and what the changes were about whether that's clinically significant to you. Because there can be something that's statistically significant, going from a behavior scale of a certain number to a certain number, but it's important that that matters to you clinically. So something that we sometimes get asked is, how does a drug get approved in multiple places? Some clinical trials are done internationally from the outset, so they will have sites in multiple countries, but more commonly, a drug will be tested and approved in one country. And then once that happens, there is the option for other regulatory agencies in other countries to conduct their own evaluation and possibly approve the drug as well. But the length of this process varies significantly, and it's not a guarantee to get approval of a drug in one country and another country. So if you are somewhere internationally and you're interested in possibly taking part in a trial or thinking about a drug, just keep in mind that it may take some time and may not be a guarantee that it will be available in another country. I would say one of the major questions that we get a lot is, what's going on with some of the completed clinical trials in Phelan-McDermid syndrome that have already occurred? What did these trials find? why were those drugs tested in PMS and what do the results really mean for our children. I have been working on a resource for this that is not finished. It's a work in progress, but we're probably going to circulate it soon, most likely on the website, on social media. And it basically includes a lot of this information that folks seem to really care about. So for example, it would include things like the rationale or the reason, and this is coming from the study team, not from us. So this is from the literature and the study team of why they've decided to use this drug. The types of measures tested, which are essentially the outcome measures, statistically significant improvements, adverse events or side effects that are related to the drug, and links to the clinical trials entry and the publications associated with these treatments. So I hope this is helpful, and all this information is available publicly on different places, but I just wanted to consolidate it and present the information in a way that you all could really use. So I hope it's helpful to you. For current clinical trials in Phelan-McDermid syndrome, please see clinicaltrials.gov and search on Phelan-McDermid syndrome under the condition. And if you click the study title, you'll get a ton of information on inclusion, exclusion criteria, locations, how the drug is administered, and contact information. So on the next couple of slides, there's a lot of information, and I don't have time to go into it all today, and Dr. Berry Kravis might talk about some of it as well. But I wanted to give really, really brief summaries of what's currently out there right now and being recruiting right now. This is not, in any way promoting that you should join these studies. It's just giving you a little bit of information about generally what these studies are testing, and also places where you can find more information. We often get questions about what could this possibly even help in PMS? And so if you look at some of these materials, you might be able to find the animal model studies and what has been shown so far, which is a guess as to what could possibly change in humans. So the first is the Neuron Pharmaceuticals trial that a lot of you have heard of that's currently recruiting. If you check out a lot of Neuron's materials, the drug is essentially designed to regulate or increase the amount of insulin-like growth factor in the brain, which is a growth factor that can have an impact in multiple tissues, but is thought to lead to better maturation of neurons and better communication between neurons. And we have some information about where to find more information on that. Lithium is not a newly designed drug. This is a repurposed drug that has been approved in the US to treat bipolar disorder and possibly other psychiatric disorders. And there have been some case studies that have shown some improvement in neuropsychiatric illness and PMS, in catatonia and with some irritability and aggression. But this is not always what is recommended depending on the person and the symptoms. I should also note that I'm not mentioning any possible adverse effects. Always before you join a clinical trial, please read the full informed consent, which will include information such as that. There's another study in Italy, which is focused on supplements, which are also publicly available. Q10, ubiquinol, and vitamins. This is based on the idea that in autism and neurodevelopmental disorders in general, there may be a defect in energy production in cells. So these supplements are hypothesized to increase energy output and antioxidant capabilities. I'm also moving through this very fast, so this is all recorded, so feel free to just pause in the recording and check out these things. Growth hormone is on my list of completed trials, and I have that information summarized because a phase was already completed, but now another phase is starting at Mount Sinai. It has been approved for genetic disorders prior, so the safety is relatively nailed down. And it's thought to have a number of different improvements based off of case studies, and it can also increase insulin-like growth factor 1. There's also a behavioral intervention that's occurring in North Carolina that has focused on not just PMS, but a bunch of other disorders within the first year of life that focuses on parent-child interaction in major common areas such as communication, visual attention, and motor skills. And finally, there's a drug that is not, the trial is not currently recruiting. It finished phase 2 and the results have not yet been published, but the second that they are, we will disseminate them to you all. And this drug is designed to inhibit a pathway that may be increased in PMS and specifically with epilepsy. So what is our role in clinical trials? First is just to be aware of the current technologies and future technologies that are on the horizon. Something that we're currently doing is completely revamping the science and medicine portion of our website and including a roadmap in science. And that will not only include, different stages of research and what we're interested in there, but also, a list of treatments that are either current or upcoming. Just to demonstrate that there's a vision in this area, and this can include things like future treatments that are not in trials yet, such as gene therapy or antisense oligonucleotides and PMS. We also are networking with pharmaceutical companies in these spaces, either at conferences or online meetings. And typically these things are proprietary, so you may not know about it right away, but the second that they can submit press releases that they're interested in Phelan-McDermid syndrome, we will let you know. We can provide data with them to help them understand if we are a good fit for them. And we're interested and invested in facilitating the development of more outcome measures that are most relevant to families. A lot of this is done through the natural history study and through a lot of the medical professionals that you know well. And we are just here to support that and increase the number of measures that are relevant to Phelan-McDermid syndrome, because not all measures really capture what's important to you all and the full range of that. Right now, we're really, as a foundation, we're really interested in GI and in communication as ways that we could help facilitate these things, but we're interested in more in the future. We represent you and we edit materials going out to you from pharmaceutical companies. And we also have a really extensive review process of any study that we recruit you all for. So staff reviews it. We have a family committee that reviews it. The scientific and medical advisories review it before we circulate everything to you through a variety of ways. And occasionally we also can facilitate travel and lodging. We don't directly fund these things as a foundation for clinical trials, but for instance, the Neuron trial, they are funding travel and lodging, and we can help disperse that. So I talked a lot about how you can learn about clinical trials throughout here, but you can see clinicaltrials.gov and search for the syndrome and check your eligibility and contact the study organizers. You will also be contacted directly by us if you are part of the PMSF membership or the PMS Data Hub, and feel free to reply that you do not want to receive these things. And we also post these things publicly on our website, in the newsletter, and on social media. I don't want to get too much into this slide because Dr. Berry Kravis might talk a little bit about it, but feel free to pause here if you're watching the recording. And the number one thing I want to mention is to always read through the informed consent document very carefully before you enroll in a trial. These things can usually be pretty long, but they'll include information about risks, about privacy, or about possible health concerns, any previous safety trials that were done. They'll let you know what the possible risks are. And last, I just wanted to mention that we're always open to your feedback. If you have suggestions regarding our role, our communication of what's going on, and what resources would be most helpful, please contact us at info@pmsf.org or me directly at kate@pmsf.org. All right, so thank you all. And with that, I I will just give a reminder that we will not be taking questions until the end, and I will introduce Dr. Berry Kravis without further ado. So Dr. Berry Kravis is a neurologist and professor of pediatrics, neurological sciences, and biochemistry at Rush University Medical Center. She's done foundational work in the characterization of Fragile X syndrome and has expanded her work to many other disorders such as Down syndrome, Rett syndrome, ASD, and of course, PMS. She's done extensive work in clinical research in many of the areas we're discussing today, such as establishing outcome measures, natural history, and design of many clinical trials. She's on advisory boards for numerous research foundations, including being active on PMSF's Medical Advisory Committee. She has an active role in helping families day to day, including seeing patients, being part of the neuropsych consult group, taking part in things like this, we can't thank her enough, and leading the Rush site of the natural history study. So thank you for being here with us. We're so glad to have you.
Okay, thank you, Kate. I'm going to share my screen. And
yes, it looks great.
okay. All right, good. So I'm going to talk about clinical trials in PMS, and some of this could easily be clinical trials in Rett syndrome or clinical trials in Fragile X, because it's all kind of generally the same concepts. And having been through a lot of trials, we've run into various things that hopefully I'm going to share with you. Okay, so what kinds of concerns do we have when we're doing a clinical trial in neurodevelopmental disorders like PMS? What are the challenges to running a trial that face both the families and the clinicians that are running the trial? Well, first of all, recruitment. Our family is going to be interested in being in the trial. And can we recruit patients without being coercive? And we don't want to tell families that something's going to happen that doesn't. And can we alleviate worries about side effects? Understanding, will families understand the difference between a trial and clinical care? And I can tell you there are a lot of situations where this gets very blurred, and really the duties that the families have as a trial participant, in that things are not just as loose as they are in clinic. You have to do everything exactly the way the trial is supposed to be. Retention, we'll be able to keep families in the trial until the end, even though the trial may be very demanding. What are the best assessments to do? We don't want the testing to be too hard on the patient, and we don't want to burn the families out with endless questionnaires or anything. So we want to get meaningful data, but we also have to, get a fair amount of data to really understand how the drug is working. Sometimes, we know this happens in Fragile X, or it did in the past, families will alter their reporting because they kind of figure out what you have to, like how severe you have to be in a certain area to get in the trial. And then this masks results at the end of the trial and is really problematic in terms of potentially making the trial not work. And then maintaining blinding and avoiding bias. And this is something we learned about when Facebook and social media got really popular, sort of around 2008, 2010, when families started to give away a lot of information on social media about what was happening to the patient in the trial. And that can be unblinding and is also problematic. So in terms of running a trial, we want to make sure that recruitment is done in such a way that the PI is available, the investigator is there to talk to the patient, and the family can be supportive and explain things. Usually we try to spend as long as needed, and give families the reasons for the trial and what is the big picture and what's going on, what are we trying to do in the long run. And then there should be an easy route to reach someone, because if you have a major concern or a side effect on a trial, you need to be able to reach someone right away. And so we try to provide numbers that you can get to people. We really work on understanding that this is not a clinical treatment, but it's an experiment, and the expectations are very different from just coming to clinic. We're studying effects in a controlled fashion, so we have to follow the protocol and all the rules, unlike clinical care, which sometimes can be quite a bit looser. And it's critical to follow these rules or procedures, or the study may fail to show that the drug is effective even when it is, or vice versa. Retention, we try to keep people in studies by making unpleasant things as easy as possible. And we haven't had too much problem in any of our various trials in Fragile X and Rett syndrome and so forth, with patients leaving the trial early because they just got burned out of the trial. But we do try to use motivators and rewards for patients to get through procedures, try to be able to provide favorite items. We try to provide schedules ahead of time that will allow families to know what their study days are going to look like. And then families should come prepared for some waiting and delays, because no matter how much we plan, we often are not perfectly on schedule. So things like iPads, toys, DVDs, et cetera, et cetera. And then we try to minimize discomfort in ways that are possible. Things like using numbing cream for blood draws, if that is regarded as beneficial for the patient, and try to anticipate things that will bother the participant and then try to minimize that as much as we can. So other solutions, when the assessments, some of our assessments require the patient to perform, and if they aren't, don't really love doing performance-based tests like cognitive tests, one tries to find the best time of day to administer those and use examiners that know how to approach the patient. But you really need very consistent methods and trainings for interviewers, assessors, and even for families that are filling out questionnaires or doing assessments in a trial. We always use breaks, reinforcers, and rewards when doing direct performance-based tests. And then we want to make sure we get accurate assessments, as well as optimizing the administration of the assessments. So it's important, for families not to really report accurately what kinds of symptoms the patient is having. It helps sometimes if the families don't really know too much about what the entry criteria are and how they're reached. And I would just say to any family member who thinks they've figured out a scoring system to get into a trial, don't tell other families, because this is not helping anyone in the long term. We've had direct experience with this in Fragile X. It's important to maintain blinding in a blinded trial, and even in an unblinded trial, it's important not to report things that are going on with the drug so as not to bias families in the next trial. And so don't tell other families about side effects or positives, especially if you're in a blinded trial. And if families suspect a treatment assignment because of a side effect, you have to kind of be the PI's partner and don't even tell the site staff or test it. Like if you have diarrhea and the drug's known to cause diarrhea, don't tell anyone except the PI, because then you can minimize the effects that that might have on eliminating the blind for other people on the study who are, say, doing cognitive testing, for instance. And this will be for the good of the study. So really, trials in rare neurodevelopmental disorders, including PMS, need to be a patient-caregiver partnership. And these are just some of my partners with Fragile X and even with Angelman syndrome. And we don't have to go as far as dressing up exactly like me to be cooperating in the trial, but in some cases people do that. We have some design issues in trying to create a trial that's going to work. So we have to think about, are we doing an open label trial where everyone gets drug, or placebo controlled, and how long should the placebo period be? Should the dosing be the same as when there's no disorder? Are there preclinical studies to help guide dosing? How do we do safety when the patients can't report if they're having a side effect? What's the best age to do the trial? And younger patients with a developmental disability might respond better, and there might be more kind of reversal of the disease. What's the formulation that we can use? A lot of times drugs aren't available in liquid forms, and our patients can't always swallow pills. Can we allow other medications, or will they mask the signal of the drug? What should be our best efficacy outcome measures? Some measures have strong placebo effects, and often measures are really not well developed for neurodevelopmental disorders, and including PMS. And then distance and travel is always an issue. So I'm going to talk about some of these things in a little more detail. When do we need a placebo control, and why do we need a placebo control? Well, early safety studies usually don't use a placebo, such as the Neuron study right now. If they're a phase 1/2A study, they're really looking for safety and a little bit of evidence about whether there's a signal with the drug. A phase 2B or 3, a larger study that's going to be used for drug approval, for most disorders will need a placebo comparator. And that's because there's variability in the symptoms or the course of the disease, and people may get better or worse at different times. There's a strong placebo effect on many measures. People can inflate their ratings at baseline to try to get in the trial. There's rater bias, even by us, the clinicians that are looking at patients, because of course we want everything to work. There are confounders like seizures and other medications and other symptoms that may influence the result. And then there can be test, retest, or learning effects. So we need the comparator to prove the drug is better. And so this is a situation where you have patients and they get grouped into a control and an experimental group. There's some situations in an ultra rare, highly variable disease, especially neurodegenerative disorders, where placebo may not be possible or ethical. And then the comparator could be natural history, case control, or within patient trajectories. I would say that PMS is not really in this category. So what are the possible drug designs? Well, everyone wants to get on drug who joins the trial, right? That's the first question that most families ask. So there's open label trials where everyone is on the drug. And this again is like the Neuron trial going on right now, but these cannot be used for FDA approval. They're usually early safety studies. If the trial is placebo controlled, it may have an open label extension where everyone gets the drug at the end of the placebo controlled period. If there could be a crossover design where half the people get drug and half get placebo, and then they cross over, and the people who got placebo the first period get drug the second period. This is not well received by the FDA, and usually these trials are not used for FDA approval, although may be used as an early study to identify the best kinds of outcome measures. Sometimes there can be an open label extension after a crossover design trial. The randomization ratio, what percentage of patients do you put on placebo and what on drug? And this may affect the placebo effect. Sometimes if you're only putting a quarter of patients on placebo, everybody thinks they're on drug. And so then that increases the placebo effect. So often trials are randomized 1-to-1 or sometimes 1-to-2. Recruitment is easier for crossover and extension designs because then everyone knows they're going to get the drug. And extensions are justified in later phase trials because you need to have long-term toxicity data at FDA in order to get approval. So you do your 3-month placebo control, and then you may do a year or a 2-year extension and use the extension for long-term toxicity data. How long can the extension go on? Well, if it looks like there's benefit and the program is moving forward, it will often go on until the drug is approved. And in fact, it may be that you can look at different measures in an extension that goes on for a long time, like measurable cognitive change, which can take a long time to see, but you can't really use placebo for the years that it might take to see things like cognitive change. The dose range is usually guided by preclinical studies in animals, and unless it's a repurposed drug where the doses are already established. And then you can dose escalate to the maximum, and there are various ways of doing that. You can increase the dose in an individual subject sequentially. You can have cohorts where a group gets 1 dose and then there's a safety review, and if they're safe, then the next group gets a little higher dose, and so on. You can have dose finding where you try several doses and decide which is best, like these where there's maybe a high, medium, and a low dose, or those may be compared to placebo. So there are various different ways of doing that. Or you can have dose escalation, but also a flexible design where you can adjust back if a patient has side effects because the patient may be doing better on a lower dose than a higher dose. And that kind of operates like the way we use medication in clinic. Safety is always challenging in cognitive disorders and neurodevelopmental disorders because the patients may not be able to report their symptoms, and sometimes the patient gives kind of random answers and don't really tell you the answer to questions about whether they're having a side effect or not. And so you need to rely on the families who really know the patient best. And families should be informed about possible safety issues. And we do this in the informed consent document. We also go over often some of the things that may have happened in animal studies and from phase 1 studies in normal volunteers so that you know kind of what to watch for. There are these comprehensive screens, side effect screens that have been developed for developmentally disabled populations like the SMRF, but these are very time intensive, and every visit you have to go through this list of like 100 questions. And it's usually more efficient just to ask about general problems and then kind of go over problems that you know might be an issue for the drug or for the patient. The age of participants is always something that we argue about. We all believe that the drug is probably going to work best if you start it when you're younger, if you have a developmental disability, but then there are safety concerns. We often have to work down from older patients because of FDA concerns about safety. And then the pharmacokinetics can be different at different ages. So in other words, the drug levels may be higher or lower, the metabolism may be different in a child versus in an adult. And so we have to have pharmacokinetics in children in order to move forward into a big trial, but that balance of that is that lots of blood draws can often be difficult for patients with these disorders. We try to get to kids as quickly as possible usually, but we have to satisfy certain safety concerns. Recruitment is often better at younger age levels because sometimes we're trying to recruit adults, they're in group homes and it may be harder to arrange for all the study visits. And assessment may be better at younger age levels because kids are still in school and you can get a better handle on their functioning, but you can certainly have cooperation issues with younger kids. There's always this question about how many baseline medications can you allow in a trial without blunting the effect of the drug. And so the purest trial, of course, you don't allow anybody to be on medications that impact the central nervous system. But of course, this makes it nearly impossible to recruit in a developmental disability where there's a lot of behavioral issues or seizures or sleep problems. And so if we need the patient to have a certain amount of symptoms to qualify, then they're already going to be on medications, and we don't want them to deteriorate if we have to take them off medications to be in the trial. This is kind of what used to be done in trials in autism spectrum disorders. And do we really want to see improvement in their worst baseline, or do we want to find out if the drug we're testing can improve over optimal current treatment? So right now, most neurodevelopmental disorder trials will allow a certain number of baseline medications for behavior or seizures. Outcome measures has been a gigantic hurdle in neurodevelopmental disorder trials. And often we need to test a broad functional range. Sometimes we can have the range of functioning in a disease, and this is actually true of PMS, can be all the way from patients who function at a very low age level to patients who are functioning sort of just below normal. And it's very hard to cross that entire range of cognitive or language functioning because there just aren't measures that have that broad of a spectrum. And few measures are validated for special populations. It's not clear how well some of these measures actually get at the core deficits. We often administer questionnaires to people with Fragile X or Rett or Angelman syndrome, and they say, that was an interesting questionnaire, but it doesn't really assess anything that's my problem. So that can be an issue. Cooperativity level can be an issue for performance-based testing. And we want to measure things that are important, measure change in the disease, and that are clinically important to families. So often we do a study where we ask families what are the most important things and kind of rank those. Sometimes a composite measure that looks across multiple different manifestations of a disease can be very useful in a trial. And often we need to design special measures like disease-specific clinician global impressions or rating scales, because no standardized measure that's already out there actually captures the full manifestations of the disease. And Neuron did a lot of work on this to try to create this special clinician global rating and PMS rating scale that captures elements of PMS because really there was nothing that naturally did that in terms of outcomes that were available. Travel is always an issue. People are traveling all over the country often for rare disease trials. Frequently, we have to have tests that can't be done elsewhere. Sometimes we can do home visits, especially if we just need blood tests or EKGs or something. And then we can have safety visits with the home nurse and do video conferencing with the physician for assessments. And this actually increased a lot during the COVID-19 pandemic. And now most trials are trying to incorporate these wherever possible. And then it's helpful just to have more sites that are specialty sites that see the patients so that there can be a trial network when clinical trials become available. Travel in an industry-sponsored trials, travel is almost always paid by the sponsor. In some federally funded trials, travel is also paid, but there may be budget limits because NIH grants are not, they don't have as much money as industry does. Drug formulation is always something that's discussed. In the Neuron trial, it is a liquid, but oftentimes it's easier to get pills of the drug than a liquid. Some participants can't swallow pills. So it's really important whether the formulation can be crushed or dumped out, which may depend on the taste. And if pharmacokinetics was done when the pill was actually crushed and given to the patient, then you can use it this way. And then it's way easier to often give to the patient. Younger kids are less likely to be able to swallow pills, obviously. So when you go to those trials in younger kids, you have to be prepared for some kind of a formulation that won't require pill swallowing. And so again, crushing or using sprinkles or something often best. Some drugs, and particularly gene therapy, may not cross the blood-brain barrier well and have to be given directly into the CSF by either a spinal tap, shown here, where we inject a drug into this patient, not a PMS patient, into this patient for their intrathecal therapy, or in this patient who actually has Batten's disease, and we're giving his enzyme directly into the brain through a reservoir, through a port there. So sometimes that is the only way to deliver the drug to the brain. So I think, you know, we discussed prior trials in PMS, and there have been a couple of IGF-1 trials run by Alex Kolovzon at Mount Sinai, and a combined analysis that he published recently suggested improvement, suggested it didn't meet the primary outcome of social avoidance improvement, but did show some improvement in sensory reactivity, hyperactivity, and repetitive behavior. The oxytocin trial didn't really show any benefit over placebo. We don't know the results of the AMO trial, as was mentioned. And there was an intranasal insulin trial some years ago in the Netherlands, which suggested that, so they tracked the rate of development for every 6 months for a year, and then randomized patients to intranasal insulin or placebo. And it looked like the insulin group was showing an increase in the rate of developmental functioning relative to their previous baseline by 0.4 to 1.4 months over placebo, but the effect wasn't statistically significant. So again, we don't have absolute proof that that was a real effect. All right, I'm going to talk now about trials, the trial that's ongoing here in the USA, which is the Neuron study of NNZ-2591. And NNZ is related to trofinetide. So trofinetide is a synthetic analog of cyclic glycine proline, which is a molecule, a chemical that's actually part of IGF-1. It's a peptide that occurs naturally in the brain and has had positive studies. And so trofinetide has had positive studies in both Rett and Fragile X syndrome, with now a positive phase 3 trial that's going for a new drug application and for approval in Rett syndrome. NNZ-2591 is a more stable. One of the biggest problems with trofinetide is you have to administer it in a giant amount of liquid, and it doesn't taste good, especially when you concentrate it and put it in less liquid. So one of the big advantages of NNZ-2591 is that it's more stable, it's more orally bioavailable. In other words, it gets through the stomach better, and it crosses the blood-brain barrier better relative to trofinetide. So this is a better drug than trofinetide. The CGP regulates the availability of IGF-1 by a competitive binding to the IGF binding protein 3. And so it regulates the amount of IGF-1 that's available to bind to the receptor. It appears to circumvent the SHANK3 deficiency in a variety of ways, like reducing the overactivity of this MAP-ERK signaling pathway, restoring depressed expression or bioavailability of IGF-1, rescuing function of microglia that maintains synapses, rescuing the dendritic morphology, or the shape of the dendrites that are connecting with each other in the brain, of the nerve tips that are making connections, stimulating protein synthesis and excitatory synapses, and decreasing inflammation also. The effects of this drug appear to be state dependent, so they correct the impairment, but they don't impact normal functioning or the normal cell. In the PMS mouse, NNZ-2591 improved deficits in in a huge list of things that I'm not going to name off all here, but lots of things that were behavioral tasks and motor tasks and seizures and anxiety and molecular things like ERK signaling and the shape of the neurons. So the phase 2 study of NNZ-2591 in PMS is going to include only 20 participants with PMS, because this is an early phase safety study with at least moderately severe disease. They have to be aged 3 to 12 and weigh at least 12 kilograms, which is like 26 or 27 pounds. They have to be more than that. It's open label, so everyone gets drug, but it doesn't have an extension. So there's a 4 to 6 week screening period to determine eligibility. You have to be healthy. You can't have kidney, liver, or eye disease. Most medications for behavior and seizures and things are allowed. You can have up to 3 meds for behavior, and all of the medications have to be stable. And then there's 13 weeks of treatment. And then after that, you have to come off of it because they don't have enough long-term toxicity data to treat people for a long time. Part of the safety data in this trial will contribute to future trials where more long-term treatment can potentially be done. And they'll collect more data in the animal models on long-term safety. So during the study, we'll be focusing on kidney, liver, and eye safety. And then we'll also be collecting a large number of efficacy measures that include performance tests like the Mullen, which is a developmental test, the Vineland, which is a clinician interview where we talk about level of function, and then different interviews where we talk about the patient's functioning in different areas, the CGIs and the PMS rating scale that were created by Neuron for this trial. The most important concerns of the caregiver: what are your worst problems? Let's rate those during the trial. And then other standardized measures like the aberrant behavior checklist and the sleep questionnaire, and a questionnaire about how many words the patient uses, and the ORCA, which is a speech measure, repetitive behavior scale, a caregiver global impression of how the patient is doing, and a GI inventory, as well as a quality of life inventory. So this is not a study that does not involve time. This is going to, the caregivers, families of the individuals participating in this study are going to have a lot of work to do to help move this forward. So there's a big list of measures. And when we're doing biomarkers and PK, the medicine is a liquid, as was trofinetide, and where the target dose is 12 milligrams per kilogram, and we titrate the dose up from a low dose, like I showed you in that slide, where there's this stepwise increase, and then there are DSMB reviews for dose escalation. There are 5 in-clinic visits, travel is covered. There's 6 telemedicine visits with study staff and an in-home nurse to come and get blood and vitals and stuff. There's a weekly monitoring of blood for the kidney and liver function. So if you have a patient that can't get blood draws, this is not a good study for you. We have a lot of blood draws. We measure weight, exams, vital signs, side effects, EKGs, eye exams. So we're really looking in detail at safety here. And then we have to have 3 participants with at least 2 weeks of data, age 9 to 12. And then there's a DSMB review, and then we can start enrolling the 6 to 8 age group, and then 2 weeks of data on 3 participants, age 6 to 8, and then a DSMB review, and then we can enroll age 3 to 5. We're recruiting as of like last week, I think 2 weeks ago. We actually have 2 patients enrolled now here at Chicago. If you're interested in the Neuron study, I think the Neuron website has opportunities to sign up, or you can call any of the coordinators at sites that are open listed on clinicaltrials.gov. New strategies, there are these new strategies to target the genetics of PMS. There's a strategy where if you only have one gene that's working and the other gene isn't, you can use an antisense oligonucleotide to increase the production, to produce more, to make the RNA kind of more stable so that you can make more protein. So what this strategy would do, it was increase the amount of SHANK3 available, which is really what the problem is in PMS, that we don't have enough SHANK3. And then there's also gene therapy that Jaguar has actually announced that they're working on PMS, and there may be others that are also looking at PMS. And this is a situation where you take an AAV9 or adeno-associated virus vector, and you take all the DNA from the virus out of it, and you put the healthy gene into it, and then you put the viral vector into the person. And in the case of PMS, we'd have to put it into the brain. So there are a number of issues with gene therapy. We're all very excited about gene therapy, but the things that we have to solve with respect to gene therapy are brain penetration. How many neurons are going to get more SHANK3? How many neurons can we get the vector into to make the SHANK3? The expression may be unregulated. There might be too much SHANK3 in some neurons. And is that a problem? Or does it just not matter if you have too much SHANK3? And then with the AAV vectors, we can only give the gene therapy once because there can be an immune reaction to the vector. And so patients who participate early in the trial may not get enough because they're still at a low dose, but then they can't get the drug again. And also the vector, the gene therapy may wear off over years, and we don't really know the answer to this, but then we can't dose again. Or we may guess the wrong dose and need to give more, but can't. There's some new companies that are developing these immune camouflaged vectors where you could give the drug, you could give the gene therapy twice, or if you gave a low dose and it didn't work well enough, you could give a high dose. And so that's something for the future that we expect to be very useful for gene therapy. And so really, I think a lot of these strategies that are starting for PMS are going on across many rare neurodevelopmental disorders. And so you can see that many of these neurodevelopmental disorders have formed clinic consortia and are collecting natural history and doing outcome measure studies, and are really training sites for future disease targeted trials. And I just show all of these because, you know, Fragile X and Angelman and Rett syndrome and Dravet syndrome and the sex chromosome aneuploidies and Smith-Magenis syndrome and PMS all have these clinic networks, which are going to be very useful for deploying future therapies where we really need a clinical trial network, and we need to understand natural history, and we need to optimize care for the disorder as we develop these therapies. And so that's what a lot of these clinic consortia are really set up to do. And so I would say one thing that's really exciting about being a pediatric neurologist right now is that genetic neurodevelopmental disorders are moving from being a field where we just have supportive treatments and we just follow people in clinic, but we can't do anything to target their underlying disorder, to being diseases where we have possibilities, for instance, for gene therapy, or drugs that work on synaptic connectivity, or even X chromosome reactivation in some of the X-linked diseases like Rett syndrome and Fragile X. And sometimes there's an antisense oligonucleotide that will reverse the genetics. And, you know, in PMS, we have some of these options coming out right now. And so there are many other new ways of targeting genetics, which I'm not going to list off, but I think we have a lot of work done, and we're starting to roll into an era where we may actually have treatments for these disorders. And we still have a lot to do to develop the right outcomes, to get good natural history, to find the right trial designs and development strategies. But I think we're starting to have strong hope that we will get there in the future. Okay, and that's all I am going to do here. Stop share. Okay. I think we can take questions.
Thank you, Dr. Berry Kravis. That was fantastic. I know I learned something as well. And just a reminder to everyone that our next session in science on September 8th is actually gene therapy. So if any people have any questions about gene therapy, Jaguar and Guoping Feng will be presenting on September 8th. And we are open for questions. We have about 5 minutes left in this session. Dr. Berry Kravis, I don't know if you have a few minutes to stay over or if we should try to field questions later. But
a lot of questions, I can stay a little late, yeah.
All right, so the first question, which I'm not sure that we can answer, is will NNZ-2591 be available for clinical trials to adults at some point? I don't know if we can answer for Neuron on that, but if you have any comments.
Yeah, I think the plan is always to go to all age ranges, right? I think with Neuron, what they're trying to do is capitalize on the fact that you might see change more rapidly in younger kids. And so they originally proposed the age range that they did, and the FDA, you know, kind of wanted to see the older patients get treated first and then move down. And so that's why they're set up the way they are, to go stepwise all the way down to the 3 to 5 year olds. So because we think that we might see the biggest change in young patients, that's why we're trying to focus. They're trying to focus early on the younger patients. But, you know, no one really knows what the exact difference is going to be between treating young and older patients. We don't know how much is fixed that can't be reversed. So certainly, if they have a positive result in younger patients, they will be exploring the drug in adults.
And that kind of leads into our next question, which is a big question and a good one, which is, what does it mean to rescue abnormal development if you're talking about younger patients?
Well, so what we want to do is make development as normal as possible. So early on, we have developmental delays in PMS, so the varying amounts of developmental delay in different patients. So the idea would be that the patient is delayed and they're not progressing at the rate that you would expect, right? We'd like to be able to implement the therapy and then show that now they're showing a trajectory that's better than what they had before. It might not be totally normal, but we're making developmental gains more rapidly than what was going on before. Or, if we use a placebo group, that we're making developmental gains more rapidly than the placebo group makes. And often when we're talking about very young patients and rescuing development, the main measures that we can use to assess whether the drug is helping or not would be mostly developmental measures.
Thank you. I see we have a hand raised. I'm not sure if that's a question, but by Andy Mitz. If you have a question, feel free to put it in the Q&A. And I'll pause for more questions. Usually they start trickling in.
Okay.
Question is, how much might treatment cost? Is that for a specific treatment, Andy, or just general questions about certain therapies being more expensive? This upcoming study, I suppose, for Neuron.
Well, so initially treatment is free when you're in the trial, and when you're in like an open label, like the Rett patients who are in their open label trofinetide extension, everything is covered by the company. But of course, the goal is to get FDA approval. And once FDA approval is obtained, traditionally rare disease drugs have been fairly expensive. But if there's nothing else available for your condition, it's not too difficult to get insurance approval for those because there's just nothing else to treat you with. So I would say that I would expect that anything that a company goes through the gigantic effort of trying to get a rare disease drug approved, the drug is going to be pricey. And, this is a chronic problem that we have where, companies have to pay themselves back for all the work to develop the treatment. And so they have to have things that are more expensive, and it's expensive for rare disease trials because you only have so many patients, you have to fly them all in, and because the measures, you have to use more special measures. And so, so this is a cost balance that we're kind of stuck with. But our job as the doctors to take care of the patients is to make sure that your insurance approves anything that gets approved that looks like it might benefit you.
Thank you. We have another question that is asking essentially if it's ever a hope of restoring developmental skills that were lost via regression. And I know that we can never guarantee something like that, but is there any hope there?
Well, that's an interesting question. Typically in neurodegenerative diseases like Niemann-Pick or Batten's disease, we tell people that once it's lost, it's gone. And the reason for that is that the neurons are dead. And so we can't get them back. But I don't think that's really the case in PMS. We don't have any evidence that neurons are dying. They're just not functioning. So if we could make them function better, I think theoretically you could. I think that's the reason why we see in a lot of the older patients, we see these ups and downs where there's a regression, but then they kind of bounce back, and they may not bounce back all the way to where they were before the regression, but there's a lot of ups and downs. And then sometimes there's a regression and then there's not a bounce back, but certainly there are lots of experiences with regressions that reverse. And so I think that's because the neurons are all there and somehow they just become better functional because of whatever treatment the patient is taking, or just because there's variation in functionality over time. And so what that suggests to me is that if we got the right treatment that optimized the functioning of shank, or that replaced the function of shank in the cells, that we could potentially see reversal of regression.
I think we have some hints of that also in animal models of, testing in older animals that seem to show some benefit. So that's great. Thank you for that. We have another question. Is there anything to learn from clinical studies of, similar molecules in different patient populations versus PMS, i.e. Rett or Fragile X? Is there any, burgeoning areas there that might be applicable across disorders?
Well, I think one thing is outcome measures and assessments. I mean, we have a lot of overlap in the measures that we use in these populations. And one of the things is that when we go to FDA with Fragile X and explain to them about what a certain measure is, then when we go back there with PMS, we're partly down the road because the FDA has already heard about this before. And maybe they told us to do some validity testing or some studies with the measure to prove that it's really tapping into the problems, and maybe we did that, and then that kind of opens the door for other disorders to use the measure. So I think there's some transfer of outcome measures. There's a fair amount of transfer of outcome measures from one disorder to another, and I think also there's some experiences that we have in running trials that we know are really problematic. I mean, literally Facebook and Fragile X trials happened at the same time, and so we did not anticipate that all the Fragile X families would go talk about all their experiences on Facebook, and that would actually bias one of our trials enough to potentially cause problems. And so now we from these learnings from past trials, we understand this, and we can now apply those learnings to PMS trials. So I think all the neurodevelopmental disorders, there's definitely trial design learnings that cross over between disorders, but also potentially sometimes drugs cross over between disorders. For instance, and then that's Neuron's idea. They have these drugs that improve sort of synaptic connectivity, and they've done a trial in Rett that was, that got to phase 3 and was successful, and their phase 2 in Fragile X was also positive. But it may be that their drugs are a group of drugs that work across different disorders. So some of the things that are developed for one disorder may lead to strategies for development for another disorder, for instance.
And that question is one that we get a lot also as a foundation, is, news or things coming out in other disorders, and then does this apply to us? And just as Dr. Berry Kravis just said, sometimes it might, and sometimes not so much. If it's a treatment that's particularly targeting, the underlying gene in that disorder, then maybe the methodology such as gene therapy would apply. But more commonly, things like, synaptic functioning, just as she said, could possibly apply across disorders.
Yeah. And so I think that's exactly right. And if you get, for instance, an ASO approved for a specific disorder, it's only going to work for that disorder. Like the Angelman ASOs will only work for Angelman syndrome because they're specific to that genetics. But for instance, trofinetide, which could be approved for Rett syndrome based on the phase 3 trial, we know we had a successful phase 2 in Fragile X, and one could envision Fragile X families trying to get on trofinetide. And there, the issue, I mean, the FDA doesn't care if you use drugs off label. It's the insurance companies that won't let you do it. So there, the issue is going to be whether we could get it approved or not. So it's actually better to have approvals in different specific diseases, just from the standpoint of availability and access to the drug, even though the drug may cross disorders.
Thank you. We have another question, which is there a better clinical study in the future, or a better approach for older patients, rather than using the same techniques used in younger patients? Or are there any considerations for older patients rather than just applying the same protocol?
Oh yeah, I think when protocols are designed for older patients, they will be different from what the younger patients are doing. I mean, I think that obviously there will be much more emphasis on regressions, and the outcomes may even be somewhat different. So Yeah, and even within the range that Neuron is looking at, which is a fairly young range of 3 to 12, certainly we can imagine patients with PMS that, can't all take the same measure. Like some of the patients that I follow in clinic, for instance, would be too high functioning for the ORCA, which is a language measure for people that are, nonverbal or close to nonverbal. So again, this is the challenge. And when we do an initial trial, we figure out, okay, how applicable was this? What populations is it usable in? And certainly when we would do an adult trial, we would be rethinking the outcome measures. Some things would be the same, but other things would potentially be addressing adult issues more specifically.
Thank you. I see one more question. Do trials ever include children younger than 3, or are there laws against that?
No, there are no laws against it, but usually the FDA will not allow it unless you've already tried it in older groups. And in fact, there are very few drugs that are actually approved for the 0 to 2 age range, because by the time you get down to age 2, the company has already run trials in adults and trials in adolescents and trials in kids, and they've extended it down to age 2. And doing trials in neonates, in young babies, is very risky. And so often the company won't do that. So most of the drugs that we in pediatrics use at very young age levels, we're basically using them off label because they work in patients who are older. There's some drugs that are approved all the way down to 0. But in some of the genetic diseases, for instance, there's a genetic disease that is neurodegenerative, and the neurodegeneration starts around 3 to 4 months of age. So they were able to talk the FDA into going right to age 0 because there was no value in doing the treatment if they didn't do it that young. There would be too much lost by the time they would study in older ages. So some depends on the disease, but I would guess that, in PMS we'll work our way down. And if we had a successful treatment, it's not clear that we would actually do trials down to age 0. The drug would just be used at that age.
So we are a bit over time. Maybe this could be the last question that we have here. And then if any others come in, I can send answers to the group. So this last question I think is pretty important also, which is, are there any studies using gene editing in other syndromes? I think it's important here also to mention that there's gene editing, which is different from gene replacement therapy. And that is a whole can of worms that maybe we don't have time to get into. But gene editing, I think, is less far along than gene replacement therapy. I don't know if there's any trials in neurodevelopmental disorders for gene editing, but possibly gene therapy. Do you have any comments on that, Dr. Berry Kravis?
So this is talking about CRISPR-Cas9 and editing the gene so that the mutation is edited out and a normal sequence is edited in, or editing the gene so that something changes. So I can tell you, I don't know of any gene editing trials in neurodevelopmental disorders. I know that there have been some in peripheral disorders that don't involve the brain. The problem has been you have to get these reagents to do the gene editing into enough neurons to make a difference. And you can do that just fine in tissue culture, but it's been actually hard to do in animal models like the mouse. And the mouse has a much smaller brain. And generally, if you can get to like 90% of mouse brain cells, you can get to like 10 or 20% of human brain cells. So the problem is that even in the mouse, they haven't been able to get good percentages of brain cells edited. And so that hasn't happened in the, in the human yet. Now, there are these RNA strategies in Angelman syndrome that they're doing where they're delivering the gene editing in a viral vector, like an AAV, for instance, vector, like like you would gene therapy, and then the editing reagents edit the RNA that blocks the paternal gene, and then the paternal gene can produce protein. But this is a very specific biology. They're not actually trying to edit a mutation. They're just trying to eliminate production of a certain RNA. So people are thinking about how to do these strategies. I think right now the biggest limitation is how to get it all in the brain and how to get enough neurons to be impacted by the gene editing. And certainly there's a great deal of work going on in that area, and people are very excited about it, but it just isn't quite ready for prime time yet in neurological disorders. But gene therapy is being done in neurological disorders. I mean, SMA is the best example, where they have a gene therapy for SMA that's actually approved by the FDA, but now SMA is mediated by spinal cord neurons, and so it's a little bit lower hanging fruit in order to get a gene therapy that's successful. But there also are a number of gene therapies that are moving forward for neurodegenerative diseases that are actually in trials now. So I think that gene therapy is going to be something that's generally used in the future, and then the editing strategies will come along later. One thing about gene therapy that one has to keep thinking about in neurodevelopmental disorders is the issue that what the gene therapy does is it puts the protein, it makes the cell make the protein, so then the protein is there. But in some cases, the protein has to be regulated exactly correctly. Like it's not as important to just have the protein as it is to have the protein be at the right place at the right time in the cell. And it's very hard to regulate that. That's been one of the things that we're a little worried about in terms of Fragile X gene therapy and potentially even Rett syndrome gene therapy. And, could be an issue in PMS, although it seems like Shank is a more basic protein that just needs to be there and maybe isn't regulated in quite as much detail as some of these other ones are. So it's going to be a learning experience. I would not be thinking of gene therapy as, oh, it's going to be a panacea. It's going to be a magic cure. We're going to get the gene into the cells and then everybody's going to be cured. I think we're going to have a lot to learn about how to tweak the system. And even the delivery, we are still, even though we're doing some gene therapy, we're not that good at getting it into all the cells in the brain yet. It's better than, for instance, gene editing, but it's not perfect yet. And so new vectors are going to get made. And some of these camouflage vectors are nice because one of the reasons the gene therapy the adenovirus doesn't get into all the cells in the brain is that it gets attacked by the immune system. So these immune masked vectors can potentially get into many more neurons, and so that may be a big advance that's coming in the next few years. So a lot of work to do, but we have strategies.
Thank you for that. I know it's a very complex question, and especially the camouflage coming up. I'm very interested in that. I don't know a lot about it, so that's good to hear about. It's occurring to me just from this whole presentation that it could help people to have some kind of informational sessions just on these new upcoming therapies that could edit either the genome itself or the RNA. We threw around antisense oligonucleotides a few times here, and even though there's no clinical trials in it yet, there is interest. So maybe there's something that I could explain and have an informational session for everyone if people think that would be helpful. But thank you so much, Dr. Berry Kravis. This was excellent. I'm sure that it meant a lot to the families, and we really appreciate you.
Okay, thanks. No problem. We're looking to move forward.
Yes, and we will see everyone for the gene therapy session September 8th, which has people from Jaguar and Guoping Fang. So thank you all.
Okay. All right, bye everyone.